All right, hello everyone. This is the notes for Target 6B, so make sure that you write down notes for this PowerPoint so that you can use it later on when you have to do the case study and when you have to take the test. All right, so this is the target that I'm looking to see if you can do this by the end of the unit. So for 6B, you need to construct an argument for evolution based on common ancestry and communicate how multiple lines of evidence support these explanations. So we need to look at what these lines of evidence are. When it talks about common ancestry, we're kind of talking about how different organisms relate to each other. So how is one organism related to another? Are they close related? Are they distantly related? That's what we're trying to see. Okay, so for the first piece of evidence that we're going to look at fossils. So we're going to watch this video about fossil evidence and how we use that to look at how different organisms are related to each other.
There are four major categories of evidence for evolution: fossil evidence, anatomical evidence, embryological evidence, and biochemical evidence. Let's look at fossil evidence first. Fossils are often used as evidence for evolution. They're the traces or remains of dead organisms that lived long ago. Sedimentary rock contains the most fossils and is formed from mud, sand, silt, and other fine particles. To make a fossil, first the animal needs to die and be quickly buried under the mud or silt so that no other critters will be able to break down the dead animal. Over time, the minerals in the mud and silt will replace the tissue of the animal and will later reveal a mold or cast of the long dead organism. The law of superposition is that older layers are on the bottom and younger layers of rock are on top. This allows us to relatively figure out the age of different organisms or fossils that we find in layers of rock. So an organism found below another organism would be older than the one on top. So letter Z is older than letter X in this case. Absolute dating places a numerical age range using radiometric dating, luminescence dating, dendrochronology, and amino acid dating. Some scientists prefer not to call it absolute, but chronometric or calendar dating instead, because the word absolute makes it sound much more certain and precise than it really is. It's still an estimation, like the relative dating of rocks based on the law of superposition, but it has a number to it. For example, in dendrochronology, scientists count the rings in a tree to estimate the number of years that have gone by. Differences in growth through the seasons create a ring, and it generally takes 1 year, but there can be some variation. Transitional species are intermediate species, which may be in the fossil record or could be missing from the fossil record. It's proposed that there would be evidence of species that were in the middle evolution steps from one species to another. Archaeopteryx, pictured here, is used to provide a link between the theory that birds evolved from reptiles.
Thank you.
Now we're going to start looking at the anatomical evidence.
All right, so that's where we're going to stop for fossils. There's a lot of big words in that video, so we are going to break down what you need to know on the next slide. Okay, so this is our fossil evidence. So we can see a bunch of pictures of fossils on this slide. So for your definition of a fossil, you can write down that they are preserved remains or traces of once living organisms that lived a long time ago. Sometimes they don't live a long time ago necessarily. Sometimes we can find fossils of things that are still living today, but oftentimes it is things that lived a long time ago and that are no longer living now. So what's highlighted in yellow, you can write that down. I'm going to keep talking about why we use fossils and how they help us to tell how animals are related, or excuse me, how different organisms are related to each other. So one of the reasons we look at fossils is, like I said, a lot of times the fossils that we look at are things that are no longer living. So we can't just go outside and observe the dinosaurs because there are no longer dinosaurs around. We can't tell what they ate. We can't necessarily tell what they looked like. So we can use their fossils, their bones, to kind of determine these types of things. So scientists can look at bones, they can study them, they can tell lots of different things like what they ate, what they looked like, where they lived, all of those things. So that helps us to tell, okay, how are dinosaurs related to some of the animals that are alive today? We can also look at fossils to see how different organisms have changed throughout time. So if we look at some of the animals that are alive today, we want to see, okay, where did these animals come from? How did we get the modern day elephant? How did we get the modern day cat or dog? So we can kind of go back in time using fossils from animals that are no longer living, and we can compare them to see, okay, was this animal related to the dog that we see today? Was this animal related to the cat that we see today? Could the cat that we see today have somehow evolved from the dog or the animal that we see in the past? So this is kind of why we use fossils. So if I were you, I would write down all 3 bullet points that are on this slide. Next thing we need to talk about is a transitional species. So transitional species is kind of a big word. Here is our definition for it highlighted in yellow. These are fossilized organisms that show the progression of species from one to the next. So if we look at this velociraptor, we see that it says underneath velociraptor 83 MYA. So MYA means million years ago. So we have our velociraptor, then we have our Archaeopteryx highlighted in yellow, so that lived 155 to 150 million years ago. And then we have the Uranus, which lived 120 to 110 million years ago. So if we're looking at this, our velociraptor is our earliest living animal that we're going to study. So we see velociraptor, then we see Archaeopteryx, then we see Uranus. So if we look at these pictures closely, we can see a lot of similarities between the Archaeopteryx and the velociraptor, and the Archaeopteryx and the Uranus looking bird thing. So we would call the Archaeopteryx a transitional species because this would be a species that shows the progression from this Uranus bird to the velociraptor that we see. So comparing the Archaeopteryx to the Uranus, we can see that it has feathers. It looks like it has wings still. It kind of has an elongated tail. The face shape looks similar, but we can also see a lot of similarities between the Archaeopteryx and the velociraptor. The longer tail, even though the velociraptor doesn't have wings, the arms kind of look like they'd be in the same position. The face again looks kind of similar. So we would say that the velociraptor evolved from something like the Uranus, or had a most recent common ancestor with the Uranus, and the Archaeopteryx is kind of showing this is how we know that. We can see that this animal kind of falls in between these two species, and it shows how we went from something that looks like the Uranus to something that looks like the velociraptor. All right, so the next type of evidence that we're going to look at for evolution is anatomical evidence, or evidence for anatomy. So we're going to watch this quick video, and then we'll talk about it, and then we will talk about our last 2 types of evidence.
Evolved from reptiles. Now we're going to start looking at the anatomical evidence for evolution. Homologous structures help scientists find relatedness among species. There are anatomical structures in different species that originated by heredity from a structure in the most recent common ancestor of the species. For example, if you look at the bone structure of a pterodactyl, a bat, and a bird, you'll see some basic similarities. This suggests they had a common ancestor with a similar forearm structure, perhaps very long ago. But if you look at how they use their forearms for flight, they're very different. The pterodactyl has one long finger that spans a large section of the wing, the bat has a wing between its fingers, and the bird has feathers all along the forearm. These are analogous structures because even though they have closely related functions, they do not have a common ancestor for that function. Each of these species evolved their ability to fly from a different ancestral line. Vestigial structures are another piece of evidence because the structures seem to serve no function, but resemble structures with functional roles in other organisms. An example that's commonly used is the human appendix. The appendix was thought to be a shrunken remain of the cecum that can be found in many kinds of herbivores. The idea is that it's a remnant of evolution yet to be truly removed. In 2013, however, scientists refuted the relationship between cecum size and appendix presence. The appendix is actually a housing complex for mutualistic bacteria that aid in digestion for many species, including humans. Embryology is the study of the development of embryos. The more closely related species would be similar.
All right, so we are going to break down some of those words that you heard in the video, take some notes about them, and then we will move on. All right, so for our anatomy notes, when we are defining evidence for anatomy, or what it means to look at species and determine how closely related they are based off of their anatomy. We want to focus on the similarities and differences in the body structures of different species. So I would write that down under evidence for anatomy, that first bullet point. The rule is that the more similar the body structures are, the more recent the common ancestor. And I want to clear something up here. So when we're talking about things evolving, a common one that we think of is like humans evolving from chimpanzees. We're not talking about how chimpanzees changed into humans. We're talking about how we could say humans and chimpanzees shared a more recent common ancestor. So there was an animal at some point in time, and then from that animal we got humans, and from the animal we also got chimpanzees. So we're not saying that something changed, one animal changed into another. We're saying that they shared a common ancestor, or this animal or organism that lived a long time ago, and from that common ancestor we got those two types of animals. So like I said, the more similar body structures that we have, the more recent the common ancestor. So if we say that fish and worms share a lot of similarities, they probably share a very recent common ancestor. That is not the case, but that's just an example. So we use three specific terms when we're looking at anatomical evidence. So we have homologous structures, we have analogous structures, and we have vestigial structures. So we need to talk about what each of those are. All right, first one we're going to talk about is homologous structures. So if we write down, or if we look at our definition for homologous structures, it says structures that look similar but have different functions. So I have an example over here. We have a human, a dog, a bird, and a whale. So we have a human forearm or human arm, we have a dog leg, we have a bird wing, and we have a whale fin. So if we look at these, we can kind of see similarities between them. So each of these has one long bone at the top. They have two bones in the middle that sit side by side. That's in white and red, and then they have these little bones on the wrist, so those little yellow bones, and then they have their long brown phalange bones, so those long kind of finger bones. So obviously a human arm and a whale fin do very different things. However, they kind of have similar structures, so we would call those homologous structures because they look similar, but they do very different things. When we have homologous structures like this, we would say that those animals shared a common ancestor. So we have our whale, our bird, our dog, and our human. So we would say that they share a more recent common ancestor because all of their structures look very similar. Next term is our analogous structures. So these are structures that look different but have similar functions. So we have our shark fin, our penguin fin, and our dolphin fin. So obviously all of these things are used to swim. Our shark uses their fin to swim, penguin uses their fin to swim, and a dolphin also uses their flipper to swim. So just because these structures do similar things, that does not mean that they look similar. So if we look at the bones of these different structures, of these different swimming structures, we can see that the fin, the wing, and the flipper all look very different. Their bone structures are not similar at all. So these would show that they do not share a common ancestor. So just because something does the same thing doesn't mean that it shows that they're related. That just means that they all evolved to do the same thing. So if all of these animals lived in water, they either are going to die because they can't swim, or they're going to evolve to be able to swim in water. So all of these animals happen to live in water, so all of them are able to swim using their fin, flipper, or wing, but that doesn't mean that they're related. And the last term that we're going to look at for anatomical evidence is vestigial structures. So these are structures that are present but are not needed. So this example is of like snakes who have still skeletal structures that could become legs, so they still have these bones in them, however they don't develop completely into legs. This shows that at some point in time they shared, or they had a common ancestor that would have had legs, and then over time they evolved to not have complete legs anymore, but they still kind of have some of those bones in there that were a long time ago legs. Another example that I like to use is humans who have tails. So when we are developing in our mom's womb, we have little tail structures, and eventually those get eroded away, and we obviously don't have tails, but some humans still have tails, and they either get them surgically removed or they kind of have to keep them. They don't really do anything. They're not harmful to the human. They just kind of like sit on their back, kind of like a tail in a dog or a cat would. So we don't have tails anymore, but a long time ago, at some point in time, we had a common ancestor that had a tail, and so now because of that, when we develop in our mom's womb, we have those structures, but eventually they go away. All right, the third type of evidence that we're going to look at is embryological evidence. So we'll watch this quick video and then we will talk about it and move on to our last type of evidence.
Closely related species would be similar in their earliest stages of development. These exaggerated drawings by Ernst Haeckel were used to show
We're going to go back a little bit.
closely related species aid in digestion for many species, including humans. Embryology is the study of the development of embryos. The more closely related species would be similar in their earliest stages of development. These exaggerated drawings by Ernst Haeckel were used to show similarities between early stages of an embryo among different species, including salamanders, pigs, and humans. And while they're not truly accurate, they were some of the first attempts at using embryology as evidence for evolution. Today, the stages are studied at the same point in their evolution, unlike these images. Lastly, when we read DNA and amino acids to find a quantity
All right, so when we're looking at the evidence for evolution, when we're talking about embryos, this is kind of what's the least important piece of evidence, just because there's kind of some complications in embryos. You have to make sure that you're looking at them at the same time in development. So for example, humans stay in their mother's womb for 9 months, but chickens don't stay in, doesn't take them 9 months to hatch. I don't really know how long it takes chickens to hatch, but it doesn't take 9 months. But for an elephant, it can take like a year. So you kind of have to look at it based off of developmental, not like the first week of pregnancy would look very different for an elephant and a human and a chicken. But with that, we're going to look at this embryological evidence. So when we're looking at embryological evidence, we can use an embryo, which is an unborn or unattached animal in its earliest phase. So again, a zygote in the mother's womb for humans, chicken eggs, those types of things. So when we're looking at these, we look at how similar they are at different stages, so and how long they stay similar for. So if we look at this picture, we have a fish and we have a salamander, a tortoise, a chicken, and a human, and it labels those at the bottom. So if we look at the fish and the salamander, they look pretty similar throughout most of their development. So if we look at the top is the earliest stage of development, it says early embryo. The middle is the mid stage, and then the last one is the mature embryo. So the fish and the salamander look pretty similar all the way throughout development. So from the top to the bottom, they look pretty similar. So we would say something like the fish and the salamander share a recent common ancestor. But if we look at like the fish and the human, the only stage where they look pretty similar is the top stage, so the early embryo. In the middle, they don't look the same, and then they definitely don't look the same as a mature embryo. So we would say that a fish and a human share a very distant common ancestor. So this animal, their common ancestor, lived a long time ago, which is why they don't have very many similarities anymore. But if we look at a fish and a salamander, it didn't live that long ago, which is why they have more similarities. Those animals haven't had enough time to change to be super different from each other, because remember, we start at the common ancestor, so it's like a point, and then there's two lines going off of that point. So we go from this point, the common ancestor, to a salamander, and this point from a common ancestor to a fish. Alright, so this is our last type of evidence. We're going to look at biochemical. That's just a big word for DNA. So if we remember, the DNA is what makes you you. It's what determines your hair color, your eye color, how tall you are, how quickly you metabolize food, what you look like, all of those things. So we're going to look at how scientists can use DNA to determine how different organisms are related.
unlike these images. Lastly, when we read DNA and amino acids to find a quantitative way to organize evolutionary ancestry, we're using a molecular clock. Mutations happen at a steady rate, and the more mutations there are mean a more distant relationship between the species. Fewer mutations means a more recent common ancestor. So, based on the amino acid differences between human hemoglobin and gorilla hemoglobin, gorillas are the closest relative to humans on this list, and lampreys are the furthest. Thanks for watching this episode of Teacher's Pet. Don't forget to like and
All right, so we're going to talk about that, and then we're going to talk about what our next steps are. All right, so let's look at DNA acids, or DNA evidence. So when we look at DNA, we're comparing the DNA, amino acids, and genes between organisms. So we could look at humans, we could look at chimps, we could look at humans, we could look at plants, we could look at plants, we could look at a bug. We can compare the DNA for any organism, and we can see, okay, how similar is their DNA? And the amino acids, remember, just make up DNA essentially. So how similar are they? And we want to look at similarities and differences to see, okay, are they closely related or are they distantly related? So if we can imagine, when things share more similarities, that means they shared a more recent common ancestor. So if we look at, I don't know why the chimpanzee says that there are 0 differences in amino acids to the human, but all of these are related to how different they are to humans. So we have chimpanzees, rhesus monkey, rabbit, cow, pigeon, bullfrog, fruit fly, wheat, germ, and yeast. So we can look at something like a chimpanzee, a rhesus monkey, and see that there are very little similarity, or there are very few differences between these two animals and humans, which means that we share a very recent common ancestor. Our DNA hasn't had very much time to evolve and change to make a bunch of differences, so we would say that because of that, we share a recent common ancestor. Again, this is not saying that humans evolved from chimpanzees and rhesus monkeys. It says that there was an animal at some point in time, and from that animal, humans evolved, and from that animal, chimpanzees also evolved. So they both evolved from the same animal, which is their common ancestor. But if we look at something like a fruit fly, a wheat germ, or yeast, there are lots of differences. There's 24, 37, and 42. This would show that humans and a fruit fly had a very, very distant common ancestor. It's been a long, long time since humans and a fruit fly evolved from the same common ancestor. So because of that, we would say that we're not very closely related. So we have had a long, long time to make lots of differences between our DNA. So we're not very closely related. So we want to look at similarities and differences. The more similarities or the fewer differences there are, the more common, or the more close the common ancestor is. The fewer similarities or the more differences, the more distant the common ancestor is. So we want to look at similarities and differences. So when we're looking at how useful this evidence is, I'm a little off. I think I switched those 2 slides. But when we're looking at how useful this is, we would say that it's very useful. This is the #1 thing that scientists use when they are trying to figure out how closely related different organisms are. And the reason for it is because you can look similar in your fossils, things can look similar in their fossils, things can look similar in their anatomy, embryos can look similar, but we really want to look at the DNA because the DNA is what makes you you. So the example that I kind of give is like you and your parents. You might look really similar to your parents. You might look very, very different from your parents. Regardless of if you look similar or if you look different, you are very closely related to your parents. Those are the most closely related people. Those are the people in this world that you are most closely related to, whether you like it or not. So I look pretty similar to my parents, but not everybody does. That doesn't change the fact that you are very closely related to your parents. So the same thing happens with different organisms. Just because they might necessarily not look the same or look exactly the same or function the same doesn't mean that they are not closely related. So that's why we want to look at the DNA, because looks can't always tell us everything. All right, so we need to talk about next steps. What should you be doing next? So on Canvas, you need to go to the Target 6B practice assignment, fill that out, and then you need to move on to the Target 6B assessment. And then once you are done with that, you are done with Target 6B.